in Antarctic Division. Thanks, Catherine. And I work in population and conservation genetics, environmental DNA, assorted questions around genetics in conservation management. And Catherine Gruber, who's from Uni of Sydney, is very kindly helping me wrangle logistics because I can't coordinate everything at once. I'll also just flag that the meeting's being recorded, and we hope to make it available later to others who couldn't be here. Okay, so I'm joining this meeting from the island of Lutruwita, Tasmania. And before we go any further, I would like to pay my respects to the traditional original owners of this country, the Muwinina people, and they did not survive British colonisation. So I also acknowledge and pay respects to all Tasmanian Aboriginal communities, all of whom have survived invasion and dispossession and continue to maintain identity and culture. I recognise their deep histories and their continuing connections to this country, including connections to land, sea, water and sky. And I acknowledge all other Aboriginal First Nations people who may be present today. And we're talking today a lot about biodiversity and ecosystems, and so that connection to country and land is really important for all of us, I suspect, professionally as well as personally. Okay, so thank you all for taking the time to be here today. We're all very excited to learn from our speakers and to bring people together for some conversation. I've got to thank Sean Hoban and Maggie Hunter, who hosted the first session, which I think some of you attended, and also the GeoBond Secretariat for the logistics, including the use of their Zoom. So that has made this meeting possible. And now what I'm going to do is give a little bit of an overview, if I can remember which buttons to push. Hooray, share screen, just of the GeoBond working group and the background to this meeting before we launch into some talks from our speakers who will tell you about some actual science. Okay, so the GeoBond working group has been around since, in this current version, since 2018. It was initiated following a request from the GeoBond leadership of Mike Bruford, who brought in Sean Hoban to get this group reinvigorated with the initial ideas to develop, test and improve approaches for using genetic observation data. So this is around using within species genetic diversity data to measure genetic diversity for informing policy and conservation practitioners, to facilitate scientific syntheses, to support integration of genetic management and monitoring into species recovery and reintroductions and population management and all of the other important things we do as part of wildlife conservation, and to promote the sustainable use of genetic resources for societal benefit. So this is all around a goal of working to better integrate genetic data into conservation management and policy. And this is particularly important in face of things like climate change and other pressures on many wildlife species that could affect population sizes and genetic diversity within them. So we've previously had a couple of all-hands sessions for this working group, and what we wanted to do here is to essentially review what we've achieved over the last few years and look at what working group members think is important, how we could work together in future, and what is next. So we're very happy to take ideas. I think there's a few long-term members and some much newer members of the group here, some people who probably don't really know what GeoBond is. So a little bit more info is forthcoming. So the group's co-chairs currently are myself with Sean Hoban and Maggie Hunter. And we also had a steering committee, which was particularly helpful in setting up in the early years. What we would like to do is now review the structure and, well, the people who are involved, who still has time to participate, and if there are any new people who are super keen, we'd really love to hear from you. So what I'm going to do then to put the context is provide an overview of the achievements and what we have been going to do. And for some reason, all of our objectives are number one. So they're all equally important. So the first few objectives for the first few years were around advancing better understanding of genetic diversity. And partly this was relating to essential biodiversity variables, which are summary metrics that can capture key aspects of genetic diversity for easier reporting for policy or management end users. We also wanted to work on evaluating and improving proxies of genetic data that could be calculated from existing data sets such as range size or functional traits or other indicators. We wanted to advance the ability to aggregate genetic information across species, across spatial and temporal scales, right up to sort of more macro genetic studies to enable that really big picture monitoring of genetic diversity. And then also to work on developing standards and models and resources around best practice to help build capacity and share information within the community. So this is going to be a complete whistle-stop tour of what we've done, which means there will not be a lot of detail, but some of the talks coming will outline some of this in more detail. So we kicked off in 2018, which meant by 2020 we started to have some outputs. One of our first papers of the reinvigorated working group was around developing this concept of genetic diversity indicators and defining 3 indicators for the CBD, the Convention on Biological Diversity. This was published as a paper, but also a policy brief that was translated into 8 languages. And if anyone misses any of these links, we can make them available later as well. And I should recognise here the fact that we've had this great group of international collaborators that has enabled a lot of this work to be translated into other languages. The next papers that come out in 2021 reviewed things like the state of the art in genetic monitoring and how to use both genetic and non-genetic data to inform policy. And a key part of this paper was really to demonstrate how workflows and concepts can fit together to non-genetic specialists to help provide better understanding of how to integrate genetic data. We also focus more on the Convention on Biological Diversity to see how different countries had been reporting on genetic diversity in their reports to the CBD. And incredibly, this policy brief that was linked to this paper was translated into 17 different languages, all available online, thanks to this working group. In 2022, we published our list of suggested EBVs after several years and many long meetings, going around in circles to settle on what we thought were the most important and best metrics to capture the key aspects of genetic diversity. And we also had a lot of thought in how to standardise data collection and collect data at scale for these metrics. And we formally introduced the Coalition for Conservation Genetics. So this is essentially a network of networks, if you like, where we got together with organisers of some other working groups who also focus on conservation genetics around the world to coordinate our efforts and to better collaborate. And this has been really important to help us share information and progress some of our policy-related activities as well. In 2023, we published this work which describes some of the connections between EBVs, essential biodiversity variables, and indicators. It can be really difficult for people without genetics experience to understand which metrics are related to which aspects of genetic diversity. So the idea here was to show how some of these different metrics connect and how to choose what to look at in a particular study. We also engaged with the development and improvement of the Kunming-Montreal Global Biodiversity Framework before and during the COP15 meeting, including not myself, but a delegation of others from GeoBon and the Coalition for Conservation Genetics were at the meeting and were very successful in getting genetic diversity included as a headline indicator in the Global Biodiversity Framework for all species, not just focused on agricultural species. This has been a huge kind of global team effort that we're all very proud of working on this particular topic. And so we're hoping that this will translate into better integration of genetic diversity in policy. We also, sorry, I've lost my place in my notes. And then we also were very happy to then be able to demonstrate the use of the indicators proposed. So we had a paper with 9 different countries showing how we could implement the genetic diversity indicators, and also some discussions around the balance of what we might want as scientists in terms of more detail and more technical research aspects of studies, but also the needs of policy and large-scale studies to be comparable and easily communicable. So this also has a policy brief related to it. And then we've also recently shown how genetic indicators can complement, genetic indicators based on non-genetic data can complement genetic data, and how there's a place for both in this space. So, you know, we don't always have access to genomic data or even genetic data in some contexts. And so we use the best data for the question, depending on what that question is. And then most recently, the working group has also developed some advice for countries who want to integrate genetic diversity concepts into their national biodiversity strategies and action plans. This will apparently be out soon in Bioscience, and there is a policy brief available already. So what we wanted to highlight was some of the features of this work. We've had a strong focus on breaking new ground around new tools, new ideas, and bringing people together across silos. We've really tried to be more inclusive in how we've worked together in terms of where people come from, their career stages, balancing different perspectives within the teams. And we would encourage anyone who's interested to join us. And if we have an underrepresented region of the world, for example, and you know someone who might be interested to join from that region, please do encourage them to get in touch. We also often aim at an audience who are not traditional geneticists. So communication and learning how to communicate with policy folks has been really important. And then we've tried to connect to those different audiences through things like policy briefs in other languages and sort of having attractive formats like the bookmark at the bottom of this slide here that we can hand out to people, and having training webinars as well online. So that's kind of a really quick summary of all the different things that have been going on across a huge group of people in the last few years. So the aims of this symposium then were to share and discuss some of these developments, to engage in some networking, and to try to reinvigorate the group by brainstorming some potential future directions. We're also interested in identifying future leaders within the working group, because there's going to become, naturally time for us to think about new people taking the lead. And we're really keen to hear from anyone who would like to contribute. So where could we go in the next few years? This is all open for discussion, but we could refine the recommendations on how to do genetic monitoring and how to use and interpret EBV data. We could identify and use case studies to show the utility of EBVs. Numbering is definitely out today. We could work on even better improved communications and connections among different groups, improve our networking and capacity building in genetic monitoring, which is certainly not even globally. And we could improve how we connect with policymakers at different levels. So that in continuing focus on the conventional biological diversity, but also national level or regional policy frameworks or taxon-specific policies are potential areas as well. And then we'd really like within GeoBond to better link with other working groups and biodiversity observation networks. So that is where we'd like to go in this meeting. I'm conscious I'm close to time. So if you don't already know how to join, or if you're not already a member and you'd like to join, you can follow these codes, but I've also put the links in the chat. So I am now going to leave with this big thank you to a huge number of people who will not all fit on a slide, but these are some of the key people involved in the Coalition for Conservation Genetics who've enabled this truly collaborative program of work. And we will move on to our first talks. Thank you. All right, that is my sharing done. And what we'll do is we'll have 3 talks now, and then we'll have a 15-minute discussion on anything related to any of these talks. And we'll also have a big discussion session on everything at the end. So could I please invite, and sorry, I have managed to lose my run sheet in the tabs whilst doing that. Can I please invite Jutta to share your screen, and we'll get right into your talk. So when my computer behaves, I can introduce Jutta Buschbom, who will be talking about the Digital Extended Specimen Network, integrating bio and geodiversity data infrastructures for global monitoring of genetic diversity.
Okay, thank you very much for the introduction. I hope you can see my slides. Okay.
Yes, thanks, we can.
So, welcome to the Digital Extended Specimen Network. The Global Genome Biodiversity Network and the international partners for the Digital Extended Specimen are collaborating to introduce you to the Digital Extended Specimen concept, the DES, our current work, challenges, and progress. The concept of the Digital Extended Specimen arose as an outcome of a consultation on the discourse forum of the Global Biodiversity Information Facility, in short GBIF, that was organized in 2021 by the Alliance for Biodiversity Knowledge. Why are we here? It took years of communication, so following intense discussions and negotiations, as worldwide society, we now have a global biodiversity framework under the United Nations Convention on Biological Diversity, the CBD. This global biodiversity framework, the GBF, with its associated monitoring framework, has the aim to guide us towards achieving the overarching goal of the Convention of living in harmony with nature. The GBF and its monitoring system will be guiding and defining biodiversity conservation action for the next 30 years. For the long-term conservation of biodiversity, research shows that we need to understand, protect, maintain, and recover genetic diversity as much as species and ecosystem diversity. Thereby, a challenge for us is to transition from phylogeographic insights to reliable regional resolution needed for many more detailed conservation questions. Human population genomics have shown us that to arrive at a smaller scale resolution that is appropriate for conservation action, reference datasets are needed that sufficiently represent the genetic diversity and structure within a taxon's full distribution range. Such reference datasets for the many taxa with wider distribution ranges are the results of highly collaborative endeavors and concerted action. Hence, distribution range-wide monitoring of genetic diversity requires data infrastructures that support and promote highly cooperative work processes. Natural science institutions and the collections community have the know-how, expertise, and established international networks needed for the management and preservation of specimen vouchers and DNA samples in support of basic research and conservation applications. They can provide the collection objects and data for investigation in local to international contexts and ranging from historical baselines to those from long-term monitoring time series. Moreover, the collections communities, researchers, and biogeodiversity informaticians maintain and continuously develop biogeodiversity data infrastructures. When we look at our data, the core characteristic of biogeodiversity data is that it is highly connected. Each information is linked to a wide diversity of derived and associated information across the boundaries of scientific systems. Here, the Digital Extended Specimen concept, the DES, provides the abstraction and technological basis for capturing and representing the highly interlinked nature of biogeodiversity metadata and knowledge. The starting point that initiates a DS is a physical material specimen or a digitally born and recorded observation. This material or digital object forms the basis of a DS and anchors it. The collection object gets digitized and will subsequently be standardized and wrapped into a couple of technical layers to produce a digital package called the object's digital twin. The widely recognized shape and functions of the digital twin package form the foundation for automatic, machine-actionable linking to derived and associated information, giving rise to the DES network. As DESs, biogeodiversity data are easily shared and combined with additional data of all types. Today, we have a wealth of repositories that focus on specialized data, maintain them, and make them available. They form the basis for data enrichment and quality assurance. Through the engagement of the community, we publicly share the wealth of data, information, tools, and services. In result, today, a specimen isn't a solitary object anymore that exists in isolation. Arising from all this linked data is a knowledge graph that sets collection objects into context and helps us to gain insight into the world. What are the implications of what we have been learning over the past decades from past experiences and feedback by you as members of the collection, research, and conservation communities, who are data providers and users, and what have we achieved? On the more technical side, one outcome is that we understand now that our data models need to change as one of the prerequisites for data integration and widespread linking. Such development activities are ongoing at the global level, for example, pushed forward by GBIF as global aggregator, and regionally in Australia, Europe, and North America, as well as locally within collection management systems where software providers are working on the data models. We are aware that unfortunately the DS and data linking are still processes that are limited in many ways too much to large organizations of the global north. We too often don't become aware of data model and infrastructure development driven by colleagues in Africa, Asia, or Latin America. We need to find solutions that enable and empower collaboration at eye level across the world and from local and small up to large and global. We are in close contact with the diverse and divergent provider and end-user communities that use biogeodiversity data infrastructures and want to take advantage of DS-based knowledge graphs. The feedback that we receive is that community members, no matter their background, want to be involved. They ask us to develop infrastructures and their functionality that enable their seamless participation. A keystone of the DS thus is the development of functionality that will enable bidirectional exchanges and updating, often called return tripping, or community curation. One focus of current platform and tool development is functionality that is expanding the options that data users have to annotate digital records, much like one would do material specimens. The aim is for everyone to be able to share their expertise and knowledge with data providers for the improvement and enrichment of biogeodiversity data. All of these developments and advances have the aim for the collections community and natural science collection institutions to be able to offer public services that provide objects and their extended metadata for basic research and operative conservation analysis throughout the life cycle of investigations, including genetic monitoring campaigns. Already today, GBIF, the global aggregator and repository of collection-based biogeodiversity data, openly and freely makes available 3 billion biogeodiversity records provided by over 2,300 institutions. These data support a wide range of inter- and transdisciplinary research, and today have been used in over 11,000 peer-reviewed papers. GBIF over the past years developed functionalities that links out from material sample data to INSDC, for example, to GeneBank records that store genomic data derived from the samples stored in GBIF. Global biodiversity monitoring cannot be done and achieved by anyone or any sector alone. The collections-based community's primary contribution to global monitoring focuses on the species and genetic diversity levels. Our goal is to reach out and integrate our expertise, resources, and infrastructures into one global social, technical, and data network for the monitoring of biological diversity across all three levels, from ecosystems to genetic diversity. We are inspired by the concept of a distributed federated global biodiversity observation system that was developed and is proposed by the GeoBond community. In sum, currently within the collections-based community, we are in a phase in which we are extending our local and specialist bubbles and are working on joining them up to the bubbles surrounding us. Key tasks for us as next steps are: we will continue to focus on communication and social network development, which form the foundation for coordination and capacity development within the overall community. We need to focus more and accelerate the development of the matrix or fabric between infrastructures. Needed is a shared, integrated abstract layer of terminology, ontology, and specifications, as well as data harmonization. And foremost, we face the urgent need to develop a business model that assures the continuation of existing services and the sustainability of analog to as well as digital infrastructures and tools. Finding solutions and making progress on these tasks will allow us to robustly solidify local efforts and to scale up to today's needs for global knowledge and services. We are on the road, and I hope I could give you an introduction to the DS and our current work. Please don't hesitate to contact us and reach out if you would like to become actively involved, or if you are interested in learning more about us as GGBN and international partners. You can contact any of us co-authors. Thank you very much for your interest.
Thanks so much, Jutta. That's crucial work. I've fully understand the difficulties of trying to connect all the different information about specimens. Does anyone have a question? We could squeeze one in before we move to the next talk. Or if you want time to think, we can come back to questions later as well. Take that. Oh, Paul, did you have something?
Yeah, just very quickly. Thanks for that, Jutta. Very interesting. How is that all funded? It looks overwhelming and huge.
Funding is mostly regionally. There we have GBEF, which is an international government organization, which is funded by the parties, so by countries throughout the world. And then we have regional activities like ALA, the Atlas of Living Australia, which is a governmental, has government funding. In Europe, it's DISCO, the Distributed System of Collection Facilities throughout Europe, which is also funded at the EU level. And in North America, we have IDIC-Bio, also federally funded by NSF. And so these are the main networks that we have, and they all have these federal regional funding. It is difficult to find funding at the global level to connect all the partners. That is something we are struggling with and are looking into.
Great, thanks.
Thank you both. Okay, we will move to our next speaker now, but we can come back to more questions later. So our next speaker is Kristy Mualim, who will be talking about genetic diversity loss in the Anthropocene will continue long after habitat destruction ends.
Yeah, cool. I'm excited. Can you guys see my screen?
Yes, thank you.
Okay, sick. Okay. Yeah, so hi, I'm Kristy. I'm a PhD student at Stanford, and I'm excited to talk to you guys about my work on how genetic diversity changes with habitat loss. So, as we all know, over the past couple of decades, land use and climate change has drastically changed our ecosystems. It's caused increased fires and droughts. And one big consequence of that is that we've seen a general decline in population size. So this is data from the Living Planet Index, where we see trends in where it tracks trends in population size across 21,000 populations of 5,000 species globally. And one big question is how this decline in population size actually impacts genetic diversity. So kind of part of the whole point of this working group is that luckily genetic diversity is already considered kind of a very important pillar of biodiversity conservation. It's now target 4 of the global biodiversity framework, where we're aiming to halt species extinction and protect genetic diversity. But in order to protect genetic diversity, there are a couple of open questions that we need to first answer. So one of it is how much genetic diversity still actually remains? Oh, okay. How much genetic diversity has been lost, and finally, what a reasonable target of genetic diversity to protect would be. In order to answer these questions, there's one way that we could do this. So we could sequence them all. This means sequencing every species, every individual, at every single time point. And there are already projects like Zoonomia and the Darwin Tree of Life that have been taking great strides at attempting this. But you can imagine that to sequence every species, every individual, and at every time point is a really big task. And so our lab was trying to understand if we can answer these questions by combining biodiversity theory with evolutionary genetics. So 2 years ago, we developed this genetic diversity area relationship. I will talk you through this relationship using Arabidopsis thaliana as an example. So assuming that this is Arabidopsis natural range, what we start by doing is we can sample different areas within the Arabidopsis thaliana 1001 genomes and count the number of mutations across all of these areas. And what we actually find is that as you increase the number of area explored, the number of mutations actually increases proportional to the number of area explored with a slight plateau at the very end. And so we find that sampling different areas within the Arabidopsis thaliana genome actually shows just this genetic diversity power law relationship. We were able to collate empirical data sets across 20 species, and we found that this genetic diversity area relationship was actually very generalizable, which was really nice to see. So the beauty of the genetic diversity area relationship is really in its simplicity. For a given area, you can get an estimate of the number of mutations that you would expect to find. And so what you end up getting is an instantaneous snapshot of genetic diversity at that point of time. But as we know, our ecosystems are changing. And so if you're really thinking about the conservation of, say, a population of interest, and in this case, a population of trees, if something were to happen to that population of trees and some of these individuals die, if you're thinking about conservation of this population, maybe something that is more interesting to think about is how genetic diversity actually changes across time. And if we can get a more accurate measure of genetic diversity, considering we know how much time has passed. And so in this presentation, I aim to basically answer this question of how does genetic diversity actually change in the long term? Does it actually change across different kinds of habitat losses? And once we have this established framework, how can we use this to inform global biodiversity policy? And in the interest of time, we'll really just focus on question 1 and 3. So the way we attempt to do this is actually by using continuous space simulations. So all of our simulations are all under neutral evolution. And I wanted to go over 2 terms that I'll be using a lot in this presentation, so we're all on the same page. So when I talk about short term, I'm thinking about genetic diversity measures immediately after habitat loss. And when I talk about long term, I'm thinking about 20,000 generations after habitat loss. So I'm really interested in genetic diversity measures that at equilibrium, essentially. And the way that we try to answer this question is using range loss simulations. So we term this edge contraction, where you lose habitat from one leading edge. And to orient everyone, because we'll be looking at these plots a lot, on the y-axis you have percent of genetic diversity within the population, and on the x-axis you have percent of habitat loss. And so in the short term, what we actually find is that there's a gradual change in the percent of genetic diversity, even at 90% habitat loss. So at 90% habitat loss, your population could still have 80% of its original genetic diversity, which is really optimistic. Now, this changes when you start looking at long-term trajectories, where you see a huge change in the percent of genetic diversity, especially at 90% habitat loss. So looking at the same population where you had originally thought at 90%, your population had 80% of its original genetic diversity. Give it some amount of time, that value might actually be closer to 10%. And so you can imagine that if you're interested in species conservation, using this short-term estimate might actually give you an over optimistic view of how your population is doing. And so really adding that temporal component is super important. So to talk about just a really short case study of how we can maybe use this, this is a bird called the shining honey creeper. In green is basically the natural range of this bird, and in red is the habitat loss in 2021. And it's already projected to lose more than a third of its home. So looking at very similar plots where on the y-axis is percent genetic diversity, but then on the x-axis now we're looking at time, and the colors represent 30, 50, and 90% habitat loss. What we can do is if we know how much a species or a habitat that a species has lost, we might be able to map it onto these trajectories. If we know how much time has passed since the species has lost habitat, we might be able to get a more accurate measure of genetic diversity within that species. So how does genetic diversity change in the long term? It turns out that genetic diversity loss continues long after habitat loss has halted. We then try to answer this question of whether genetic diversity trajectories will be different across different kinds of habitat loss. And the way we try to do this is using a fragmentation scenario. If anyone wants to talk to me about this later on, I'd be happy to, but we'll skip this for now. And then finally, now that we have an established framework, how can we use this framework to actually help inform global biodiversity policy? So first, we look at demographic indicators that people have traditionally used to estimate, extinction status in species. So one is the Living Planet report, and the other one is the IUCN Red List. We're going to attempt to basically get global genetic diversity predictions off of these indices. And the calculations are the same, so I'll really just focus on the Living Planet index. So using our framework that I described earlier, and then using population size declines as a proxy for habitat loss, we can then get, this is essentially a density plot of genetic diversity loss in the short term. And you can see that the distribution is fairly bimodal. You have a cluster of species that have lost 90 to 100% of their genetic diversity, and then you have another group that have lost 0 to 30%. In the long term, the loss ends up being a lot more dramatic. So you could see that the distribution has entirely shifted to the right. Species that you would have thought lost 0 to 30 are now maybe losing really a lot more. We wanted to put these values in the context of a previously proposed but now removed UN target of 90%. It was to protect 90% of genetic diversity. And using our estimates, we actually see that in the short term, half of the species have already lost 10% of genetic diversity. We estimate this to be a 12% short-term genetic diversity loss. And in the long term, this actually ends up being way more dramatic, where we're estimating that this is actually going to be a 37 long-term genetic diversity loss. And these two estimates are actually already well past the previously proposed UN target of 90%. Finally, and in the context of this group, One thing that we would also like to do is to try to think about global genetic diversity predictions using the indicators that have been proposed by this working group. So recently, indicator 1 and 2 was calculated across 9 different countries and across 20 or a dozen taxonomic groups. And we wanted to see if maybe we could use proxy area loss as by answering this question of of the proportion of populations that still exist, how many of these populations have any less than 500? And we have genetic diversity predictions for that as well. But I'd love to discuss more with everyone on this call. So to conclude, we find that genetic diversity decreases long after habitat destruction halts. This is likely going to look different when you look at species with shorter generation times as opposed to longer generation times like trees. And the final question is really thinking about how do we reconcile what we've already lost with a reasonable genetic diversity target. A lot of this work is already in like a preprint, so if anyone's interested, I implore you to check it out. And finally, I couldn't have done any of this work without support from my lab, specifically Meishi, Jeff, Clemens, Oliver, and Moy. And thank you guys for listening.
Thanks, Kristy. Thanks so much. That's quite sobering, really, isn't it? Would anybody like to ask a question at this point from Kristy? I can see little reactions popping up. Sorry, they're confusing me. I had one question quickly that I could ask you, which is it looked like you had included quite a number of different taxa in some aspects of that work. But how generalizable is it, do you think? Is it kind of the sort of approach that could be applied to pretty much any kind of, like marine, terrestrial, different taxonomic groups?
Yeah, so one part of this work that I didn't show is that we're actually trying to collect genetic data of just empirical data of like species, so anything that's like publicly available out there. And so far, we found that at least for short-term estimates, they actually follow a very similar trajectory. The hard thing is that we can't actually collect any empirical data for long-term estimates because the long-term estimates that we have is really far off into the long term. So it makes it hard to see if this is, yeah. But these are just estimates that we're hoping could be refined in the future with maybe better simulations or, other people who want to build on the work.
Great. Thanks so much, Kristy. Oh, do you have a question, Jutta?
Yeah, thanks a lot. That's really exciting. I'm wondering, so you're taking a random sampling approach of diversity over the distribution range. Have you looked into describing diversity at different time slices? Because some species have been moved around a lot, or they had a continuous distribution range at further in the past, but now are highly fragmented. And to kind of like define what is actually the driver of this continuing degradation of genetic diversity, where you would need to kind of separate the old versus the newer pattern. Have you looked into that?
Not like, yeah, not comprehensively, but we see a little bit of it in our fragmentation case where I think so all of our measures are species-wide genetic diversity, and then we see in the fragmentation case, when you wait long enough, such as in the long-term scenario, your populations actually start diverging from each other. And so in those cases, we can actually start tracking individual populations and seeing how they do. But in terms of thinking about we're just looking at like neutral genetic diversity as a whole. So like one component that would be interesting to add is to add in like adaptive or deleterious like mutations and see as populations like diverge from each other how they do. But I think your question is actually a really good point. Yeah, it's super interesting.
Thanks.
Thank you very much, Kristy. So we will now move to our third talk of this session, and this comes from Jasmine Tschan, who will talk on assessing genetic effects of direct seeding on a dry meadow plant species using combined genomic resources. Sorry, thanks, Jasmine.
Thanks for the introduction. I just start sharing my slides. Can you see them?
Yep, thank you.
Oh, thank you. So, hi, I'm Jasmine. I'm a PhD student from ETH, and I welcome you to my presentation about assessing genetic effects of direct seeding on a dry meadow plant species, in this case, the Carthusian pink, as you see it on the picture, using combined genomic resources. As we all know, biodiversity is under threat through various drivers, like, for example, habitat destruction and climate change, as we heard before. National and international authorities have seen those threats and started implementing conservation measures, like, for example, translocation or reintroduction or revegetation. As revegetation measure, often direct seeding is used, which aims to promote biodiversity, establish stepping stones, and prevent invasive species from growing. However, the seed provenances that are used for the direct seeding can vary heavily in genetic composition and fitness, and leading the outcome of the revegetation measure, which can have positive effects, like, for example, establishing new populations somewhere or boosting genetic diversity when hybridizing, but can also have drawbacks, like, for example, maladaptation or outbreeding depression when hybridizing. So we in our study focused on dry meadows, and because dry meadows are hotspots of biodiversity and habitats of national importance in Switzerland. And additionally, dry meadows have had a severe loss in the last 100 of years. So we focused on Dianthus carthusianorum, or the Carthusian pink, because it is widely used in biodiversity promotion in nature and also along infrastructure. However, it is not well documented when and where direct seeding actually occurred or what's the extent of it. So we asked ourselves, how can we actually detect direct seeding using genomic tools, and what's the extent of direct seeding? To answer that, we had a random sampling across the Swiss distribution range of the Carthusian pink and has 31 populations of whole genome sequencing that were available from the pilot study for monitoring genetic diversity in Switzerland. This pilot study actually not only looked at the Carthusian pink, but also for other species across Switzerland to establish a genetic diversity monitoring. Now, to come back to our research questions, we also asked ourselves, what are actually the effects, the genetic effects that we see from this direct seeding, and where do the seeds come from? To answer that, we had to enlarge our geographical sampling scale and add populations from outside Europe, outside Switzerland in Europe. And those were dd-red single and paired and sequenced populations from different collaborations and studies that we were able to add. To combine these different sequencing methods, we created a de novo SNP panel. However, we also had to consider potential evolutionary lineages in the Carthusian pink, because as we all know from Hewitt, there are many species that have different evolutionary lineages that meet at secondary contact zones, and Switzerland is quite close to some of those well-known contact zones. So we considered that for the Carthusian pink. And we found actually 6 different evolutionary lineages across Europe with high genomic divergence, with an FST ranging up to 0.43 within Europe, but also on a really small scale within Switzerland. Additionally, there was also a high phylogenetic divergence, so we classified these evolutionary lineages into evolutionarily significant units, or ESUs. So in total, we found 6 ESUs across Europe, 5 of these also in Switzerland. And here we already have a first hint of direct seeding when we look at these populations that do not actually originate from Switzerland, but rather from Eastern Europe. But how to detect that with other populations that are not that obvious? So to infer direct seeding, we looked at a phylogenetic tree and an admixture analysis. And first I will show you the natural populations in the tree. So these are those. And here we see just all the individuals that represent one line in the tree clustering within their population, which is one color. However, when we look at direct seeding from homogeneous seed sources, this can look quite differently, where we have just a lot of individuals from a lot of populations clustering in one huge population, the homogeneous seed source. Another way to detect direct seeding is by looking at that mixture. For example, these two populations here are mixed out of 3 ESUs. From at least one of those ESUs does not originate at the location where the population was found. So another hint of direct seeding. And last but not least, non-autochthonous populations that are not admixed, but do not originate at the location where they were found. So in total, we found that 48% of the populations were established through direct seeding, which is visible as triangles and squares on the map. And out of those, 22% were admixed between 2 to 3 ESUs. The amount of admixture per population is visible as different red to orange to yellow shapes in the map. And we also found 13% of non-autochthonous populations. Additionally, we wanted to detect the drivers and timing of direct seeding, so we looked at aerial photographs at different time frames, as we have it here, for all the populations. And on average, the drivers of the habitat modification were due to building of roads, as we have it here, and other infrastructure, like buildings, but sometimes it was even when doing, for example, river revitalizations, that then after the process there was seeding along the river. And the time since the habitat modification was on average 12 years ago. So we found quite a high percentage of seeded populations, and we know that seed provenances can differ heavily. So we asked ourselves if we see significant differences of genetic diversity across the seed provenances and ESUs. So what we see here is a box plot of the different ESUs, so each color representing one ESU. And when there is a box plot with 2 box plots from the same color, we have natural and seeded populations. So the natural individuals are drawn as circles and the seeded ones as triangles. And we do found significant differences in seed provenances and ESUs, which can occur through evolutionary history, as was potentially for this ESU the case that contains only natural populations, but could also be due to bottlenecks in seed production, because this ESU does not have any natural population, but we only had seeded populations in Switzerland. Additionally, we see a trend in loss of genetic diversity during seed production. However, this is not significant, but still it would be really important to retain as much genetic diversity as possible during seed production, because genetic diversity is essential for the resilience of the populations to adapt to environmental changes. So to sum up, combining different sequencing information can be highly beneficial to enlarge the geographical sampling scale and enable various conservation-relevant inferences. We were able to classify 6 ESUs of the Carthusian pink, 5 of those appearing in Switzerland. And direct seeding can be detected using genomic tools. We found that nearly half of the randomly sampled populations across the Dianthus carthusianorum distribution range in Switzerland were seeded on average 12 years ago, half of those were already admixed out of several ESUs, and a quarter were non-autochthonous. And this has unknown consequences on the natural populations, especially because we found significant differences in genetic diversity in the seed provenances and ESUs. So if you're doing biodiversity promotion, which is highly beneficial, but you're using seeds that you're using seeds that potentially are highly diverse, or you don't know about the seed provenances, I would advocate to use local seeds. And with that, I want to thank you for your attention. And if you have any questions, you can ask them now.
Thanks so much, Jasmine. Are there any questions first of all for Jasmine on the basis of her talk? Oh, a couple of hands. Sasha, I think you were first.
I suspect we may have the same question. So there was no evidence, as I understood, from of the outbreeding depression or any negative fitness effect of the mixture. Why would you recommend local provenancing?
Because I additionally looked at the different seed at the seeds from seed suppliers of different seed provenances in Switzerland for potential further research. And when growing those seeds, we also got hybrids of the, potential hybrids of the different seed provenances. And we had quite an effort to bring them up, actually, because we just had to reseed them, I think, 5 times to just get like 10 individuals. And that's also a reason why. And because they are really, really highly diverged, especially these 2 lineages with the FSC of 0.43, because they are as diverged as other species. I can show you another slide potentially. How does it work? Oh, here. This is a phylogenetic tree of the, oh, sorry. Well, can you see it like this as well?
Yep.
Great. Well, this is a phylogenetic tree where I have the different ESUs from the Carthusian pink. And we also have Dianthus sister species. And we have 2 well-described species that cluster within the same clade as the Carthusian pink. So they're as diverged as described species, depending on the ESU.
But wouldn't you think that species description can be arbitrary, and FSD is probably not a good, I mean, phylogenetic signal is certainly a different thing, but FSD, high FSD could arise just by drift, right?
Yeah. The other thing is that we also have this Pontedere that cluster within that are locally adapted. Another hint that, yeah, others might are probably locally adapted too, and yeah, that's just to be cautious.
Thank you.
Oh, Paul, do you want to follow up?
Do we have time?
To a 15-minute discussion. So I was going to invite, first of all, more questions for the first 3 speakers. So I think you fall into that category.
Okay, great. Jasmine, that's really great work and subject close to my heart and many people involved in this sort of area. There's actually a conservation genetics specialist group at the moment working on ESUs, and there's been a big discussion around that regarding once you've found ESUs, you know, what does that mean about what you do with them? So Sasha and I work together. So Sasha was right. My question was much the same as hers. Just broadening out from what Sasha said, whether or not local provenance would be a good thing absolutely has to start with what your conservation goal is. So if your conservation goal is to maintain 6 ESUs, then you shouldn't mix them. If your conservation goal is, I've got this ESU and I really, really love it, I don't want it to go extinct, but it's got terrible inbreeding depression or self-incompatibility problems, then you should mix it. So I both of us, I think, and many people in the field have a sort of visceral reaction to local provenance being promoted in a, you know, without caveats. It's totally dependent on what your conservation goal is.
Yes, definitely. Well, the Carthusian pink is also quite a frequent species, so potentially the inbreeding depression there is not that high yet, at least. So the conservation of the different ESUs would make sense in this species, but I definitely agree it really is species dependent. And well, this also a bit arises from the seed practices in Switzerland, because the seed suppliers aim to promote kind of regional seed use and really start doing that on a really, really small scale, an area of a few, maybe 100 kilometers at max. And so this is also a bit going in the direction of showing that it is really an effort to do it to preserve the local diversity, but also
Absolutely.
it's yeah, you don't even have to do it on such a small scale, because yeah, the ESUs are a bit bigger, and yeah, with that you don't need to preserve it on such a small scale.
Exactly. So as for some questions, depending on what your conservation goal is, there's a genetic downside to local provenance, or can be, and there can be that practical one that you just said. And I mean, we have to be really careful about how we communicate this with the people who work in the seed provenancing industry, because some of them are more sophisticated in these concepts and others are less sophisticated, and we can't overcomplicate it, but we also shouldn't undercomplicate it. And local is best can be wrong very, very often. Thank you.
Thanks, Paul. Thanks for the really good discussion there, everyone. So I'm just going to quickly recap. We've had 3 talks so far. We've looked at challenges in data infrastructure and the importance of sensible data infrastructure for connecting specimens and genetic data. We've talked about the temporal lag in detecting loss of genetic diversity and how we need to better account for this in future predictions. And we've had a nice example showing application of genomic data to understanding conservation relevant questions. So what we now have is a few minutes where we could ask questions of any of the 3 speakers so far, or any other thoughts or ideas you'd like to raise, and then we'll take a short break. So I'll throw it open to anybody who wants to ask anything relevant. Yeah, please go ahead, Jutta.
I actually would have a question to all of you. So yesterday and today, the focus was on describing what remains of genetic diversity as a foundation for protecting it. I'm actually coming from a bit of a different angle because one of my backgrounds is forest genetics, where there's a question of enforcing protection. So you want to be able to assign where timber came from. So you need to do assignment, which requires partly a different perspective. So I was wondering if people have ideas about this, opinions, perspectives on how these two perspectives can come together, or if they are totally distinct, what genetic diversity can contribute to it. Yeah.
Would anyone like to take that one as a start, or?
Sorry, I'm
Shall
not
I?
sure that I actually understood the question entirely.
So, oh, sorry, Paul, you go. I think if I understood correctly, you're asking about, rather than understanding genetic diversity to conserve it, itself, more about how understanding genetic diversity can help with things like provenancing the source of a specimen or some item that has been found. Yeah.
Yeah, it is actually not looking at a species or a lineage globally, but going into its structure and looking at it like it's kind of like the ESU approach, using it for assignment, which my experience is opens a whole new world of questions. So,
well, we have a pioneering lead exponent of that on the call, because Ollie Berry did that with excruciating detail with a grand rock skink, where he could say exactly which pile of rocks a skink came from his PhD 100 million years ago. Ollie's not taking the bait.
Ollie, you need to unmute.
I was double muted, and I was thanking you profusely for your kind words. And it was a very long time ago, so I don't think I'm equipped, other than to say, I think it's quite well established that assignments are really, depending on the sort of life history and the sort of the questions being addressed, is a really powerful way to use genetic data to assign provenance. And I guess, sorry, I missed a bunch of talks, so the other half of the context I'm not really equipped as well. I mean, I guess it relies on there being quite well demarcated units of genetic diversity that can be distinguished with these statistical approaches. I think with the rise of larger genomic data sets, where we've been able to do that, the power to do that is enhanced. I guess once you start, just from that last talk, there was some discussion of mixing stocks. I mean, that's going to be at odds perhaps with your capacity to assign provenance. I know that's a very obvious thing to say, but that's what I'm going to say in the absence of seeing most of the previous talks. I don't know if that's really getting to what you're at, Jutta, whether there's a tension between one objective to potentially maximize diversity by perhaps moving or not moving organisms around the planet, and the desire to retain very distinctive stocks so that you have the power to use provenance type tools. In some circumstances, I could see there'd be a tension there.
Certainly, as differentiation goes down, it's more difficult to assign to population level. The beauty of highly resolving genome data is if you've done the sampling right, potentially you will be putting things to individual parents rather than populations, and that can overcome that problem. But other than that, you're quite right. As things merge, it gets harder to to assignment. In general, though, assignment works super well, which I think you'll see in a little bit in Sasha's talk later on, and quite possibly some others.
Is that what you were getting at, Jutta? Yeah,
I was wondering, like, because as I understand it, the symposium is about looking forward and what the working group wants to do. And if this is a topic for the CBD that we would like to enter into the monitoring framework. And then unfortunately, I have to kind of like contradict a bit my experience with assignment, which very widely, continuously long-lived species is that it's really, really difficult if you want to get probabilities that are actually operational. So it's more a question about, is there something that the working group would like to go into or not? But I'm happy with your answer, so thank you very much.
Sasha, are you got something on the same topic?
Slightly different topic. So the question to Kristy, as just thinking about the simulations and how they show that populations, when you start detecting population loss of population diversity, it's already, huge amount, and we have no chance to stop it once it started. But can we reverse it by mixing? And I was just wondering if simulations that Kristy did included or will include some sort of, mixing populations back and seeing how you can restore genetic diversity by reversing the pattern of decline through, for example, augmented genetic diversity.
Yeah, that's a great question. We did some preliminary analysis of doing I guess relocation. So we would like pick an individual from the furthest edge of the habitat and then try to just move them to a habitat that was destroyed. And eventually you do end up getting restoration of genetic diversity, but it does take a really long time also. Yeah, in the same way that the genetic diversity loss took a long time, the restoration also takes a really long time. And at least in our simulations, we still can't differentiate between whether that genetic diversity is at all helpful for adaptation in any way, or if it's just an accumulation of new mutations.
And are you planning, yes, you said you are planning to introduce some selection in your models too, right?
Yeah. That would be the dream.
Cool. Thank you.
Thanks, everyone. So I'm going to just note the time that, according to my piece of paper, which is 4:15 in Eastern Australian time, it is break time. So we get a whole 30-minute break to, I don't know what meal it is at your time of day, but for me, it's probably dinner. We've got 3 more talks and then another discussion session to wrap up after the break. You're welcome to just leave Zoom open and, just mute and turn your camera off and pick back up again. So at 4:45 p.m. Australian Eastern Time, which is, equivalent time for wherever you are in the world, go get a cup of tea or a coffee or a snack or something, and we'll see you back shortly. Thanks.
Thank you.
Okay. Excellent. Okay, thanks. So welcome back after the break. So our next speaker, if I've got my order correct, is Alexandra Pavlova. Sasha, do you want to share your slides? And we are going to hear about developing a metapopulation genetic management approach for persistence of a species threatened by fragmentation. So we're going into fish world.
That's it. Good.
Yes.
Oh, excellent. Hello, everyone. My name is Sasha Pavlova. I'm an evolutionary biologist and a senior researcher at Monash University. I'd like to acknowledge the traditional owners of country throughout Australia, recognize their continuous connection to land, waters, and culture, and pay my respect to the elders past, present, and emerging. The work I will present today is the result of many years of collaboration between university researchers, including my colleagues, and wildlife managers, whose names are here in green. Genetic diversity, as we know, underpins diversity of phenotypes and thus population ability to persist and adapt to changing environments. Habitat loss and fragmentation limit gene flow, leading to isolation, and small and isolated populations rapidly lose genetic diversity, becoming inbred and develop inbreeding depression. These genetic problems elevate extinction risk. But augmented gene flow presents a natural solution to these problems. Gene flow can decrease inbreeding, increase population fitness, and provide variation for building new adaptation. There are published guidelines on how best to augment gene flow. It is considered safe to connect populations that are diverged recently, similarly adapted, and have been in gene flow in the past. Connecting populations that diverged long ago, are differently adapted, and have different number of chromosomes may be more risky. However, while weighing benefits and risks of gene flow, the risk of doing nothing must be also considered. In some cases, doing nothing can doom populations to extinction. Macquarie perch is an Australian freshwater fish. When it was first discovered 200 years ago, it was widespread and common. Pink color on this map shows former species range, and this 100-year-old picture shows a single-day catch from a population that is now extinct. After rivers were dammed for agriculture and other human use, Macquarie perch distribution was reduced to a few isolated remnant populations, shown here as red squiggles. This species is currently recognized as endangered. 12 years ago, we assessed the state of Macquarie perch genetic diversity and connectivity. Our results showed that currently isolated populations were historically connected by gene flow. Now populations are strongly differentiated and losing ancestral genetic diversity through genetic drift. As a result, most populations are under risk of extinction through inbreeding and reduced adaptive potential. And in some populations, extinction due to inbreeding depression appears very likely. To avoid further extinctions, immediate actions were needed to restore gene flow. So we recommended reconnecting populations, using translocations within catchments, conducting captive breeding trials to test for outbreeding depression of between catchment crosses, and releasing the offspring of such crosses if they turn out to be viable, fertile, and show no signs of lowered fitness when compared to within catchment crosses. These recommendations have been adopted in the National Recovery Plan. Mixing fish from different populations has since occurred in several populations, and 3 populations were studied, genetically monitored, yielding important information for future management. And I will tell you a little bit about one of these cases, which was the case of successful reestablishment of extinct population in the Ovens River by mixing 2 sources. One source was historically a mixed translocated Yarra population, shown here in yellow. The other relatively large remnant population from Lake Dartmouth, shown here in black. In addition to translocation and stocking from these sources, captive-bred mixed ancestry offspring were also stocked. We found that offspring of Yarra and mixed pairs had best survival rates after stocking. Most locally born fish were of Yarra or mixed ancestry. And moreover, more recent monitoring of the ovens populations revealed that fish that colonized local tributaries were also of Yarra and not mixed ancestry. Overall, genetically admixed riverine individuals appear to have a superior fitness compared to those of lake-only ancestry. These results suggest that using multiple sources can benefit population recovery. Also, using mixed parents and captive breeding can increase offspring fitness. And finally, river sources may be better suited to translocations to rivers than lake sources. This may be because river fish are under stronger purifying selection or directional selection for river environment. In a recent study, population modeling was applied to estimate the effectiveness of several management interventions while considering the effect of climate change on Macquarie perch populations. This study found that risk of population extinctions is lower when augmented gene flow is included as one of the interventions, and when population is located in a climatically stable location. Alarmingly, all Macquarie perch populations included in that study appear to be vulnerable to climate change, especially increasing number of catastrophic fires and droughts. Thus, reintroduction of Macquarie perch to climate-secure location was suggested to preserve species-wise genetic diversity. We now extend this approach to a metapopulation approach, where we suggest to proactively mix different sources to prevent further loss of genetic diversity by drift and fuel adaptation by augmenting genetic diversity. To select best combination of sources for establishing a population in a climate-secure location, we can use simulations in metapop2. Here, we estimated proportion of individuals from each population that maximizes heterozygosity of a new population, or that maximizes the total number of alleles in a new population. These two strategies could be quite different. To maximize heterozygosity, we would use sources with high-frequency alleles that are rare in the rest of the pool. To maximize the number of alleles, contribution from all population is needed, or with more individuals from sources with many low-frequency alleles that are rare in the rest of the pool. The final choice of the strategy could be made depending on our conservation aim. To rapidly reduce inbreeding, maximizing heterozygosity may be the best strategy, because heterozygosity is closely linked to inbreeding. But when the aim is to preserve adaptive potential of the whole species, maximizing the number of alleles may be a better strategy. The two approaches are not mutually exclusive. When new population is first established, it is necessarily small. Thus, maximizing heterozygosity could work best in the beginning of translocations to avoid inbreeding in the future. When population grows, migrants from other populations could be added to increase the number of alleles it harbors. Once established and self-sustaining, such population could be used as a source to supplement small remnant populations in perpetuity. To calculate baseline values of population genetic diversity for future monitoring, we created a pipeline called GEDI. This pipeline combines existing genomic tools to calculate unbiased estimates of genetic diversity from reduced representation sequencing data. We capitalized on the new script called PIAFCA, which accounts for missing or low coverage sites. It also incorporates variation at polyallelic sites and scales heterozygosity at variable sites by the total number of sites that are assessed. The output is individual heterozygosity population nucleotide diversity and between population sequence divergence. Estimates of nucleotide diversity obtained with this pipeline are expected to be comparable across populations, species, and timescales, and thus suitable for monitoring change in genetic diversity. We compared estimates of unbiased heterozygosity and nucleotide diversity calculated using our pipeline with SNP heterozygosity and allelic richness. Individual SNP heterozygosity and unbiased heterozygosity were highly correlated, although some population-specific biases were apparent. And nucleotide diversity, while generally correlated with allelic richness, was much better reflecting increasing genetic diversity of the Avans River after combining two genetic sources of migrants. This population had twice the nucleotide diversity value of the either source population. So in summary, we promote a proactive metapopulation approach to reconnect populations that became fragmented through human activities. The aim of such approach is to preserve and restore fitness and adaptive potential of the whole species. While smallest populations may need to be specifically managed for maximizing heterozygosity to alleviate inbreeding, all populations need to be genetically reconnected to ensure populations can adapt to future environmental changes. Creating populations in climate-secure locations can help preserve existing genetic diversity of the species. It is important to continue monitoring genetically augmented populations and measure success of genetic augmentation. Augmentation can be considered successful when genetic diversity increases, when differentiation between populations decreases, and currently private alleles become shared among populations. Interventions that could be changed based on the results of this monitoring, and this is the basis of adaptive genetic management. With that, I'd like to thank all my collaborators, colleagues, wildlife managers, and many agencies who funded this work across many years. And thank you for listening.
Thanks, Sasha. That's great. Can I ask if anyone has a question for Sasha? And I think I can see a hand. Is that Yasmin's hand? Sorry, I can't see everybody at the moment.
Yes, it is my hand. I wanted thank you for the presentation. It was really interesting. And I wanted to ask if you looked at genetic load of the populations as well.
This is a good question. So we are planning to do that next using whole genome sequencing data, because at the moment we're just having this reduced representation data. And our species, Macquarie perch in particular, is very low diversity. So in a way, it's just not much information. So we are working on obtaining whole genome sequencing data, and we'll look into it. Definitely a good question.
Sasha, there's a request in the chat to share, I've lost it, the 12th slide.
Share slide 12. Hold on. Summary.
Summary slide.
How do I do that? Copy paste.
Or even, I guess, just to show it on the screen. But yeah. So for anyone who missed at the start, we are recording these talks, and I don't yet know when we will get them available, but the aim is to make them available for people to watch again if needed to. So yeah. Thanks, Sasha. Does anyone else have a quick, we do have a quick question. Yep, Jutta, please go ahead.
It's really exciting to see how well you know your species and your populations. I was wondering, over what kind of time scale have you been studying these populations, and how many people were involved or are involved in this? Thinking about scaling up to the number of species we have and so on? Is this an approach that can be scaled?
Well, I think certainly genetic management and moving individuals can be scaled, because managers do manage their populations all the time. So this is just additional tool that managers can be using, and not many of them actually know about them and know about the reasons for using them, right? So it is not actually very expensive to move individuals or plants or seeds, and the benefit to fitness can be really huge. In terms of how many individuals, people involved in this work, I would say over 10 years, actually it's closer to 20 years, 15 years, there would be maybe 30 people directly involved into all kinds of data collection and genetic research and population monitoring across the whole species range. But this is not about the number of people, this is about connecting to individual managers who are already doing this work, right? Because we're not specifically creating this project to do this work. We are linking universities and managers, so we all can co-design management interventions based on the best possible knowledge. Managers are usually very good listeners and like to make decisions that is based on science, because in many cases they have to make decisions anyway, and when they have some data, it's just really a pleasant job for them. Paul, you want to comment?
Just quickly, it's a great question. Upscaling super important. The groups like GeoBon, as you will probably know, have been doing things that are very pragmatic. So being able to make decisions on non-genetic marker-based genetic data and genetic proxies has been very important. So Sasha's presented that very, very detailed case on a fish, and then there's, we have an equally detailed one on, well, 2 birds and a mammal and a plant, and over the world there are dozens of very detailed examples. But if you take those as case studies, you can then apply the principles, which we've done, for example, in the state of Victoria. We did a kind of genetic State of the Union sort of report for 1,150 species. And like your organisation, people are very mindful of the need to be able to produce things at scale.
Thanks, Paul. Thanks, Sasha. Okay, so I'm going to move us on, but we can come back to more questions in this discussion at the end if we want to. Our next speaker is Isaac Overcast, and unfortunately at the very last minute, Isaac hasn't been able to attend, but we do have a recording that was made especially for us. Now, Catherine, are you going to play this or am I going to play this?
I can do it if you like.
Okay, cool. I trust your computer slightly more than mine. So we'll just all watch this, and then obviously we can't ask questions, but I'm sure we can contact Isaac if needed later.
Yeah, just give me a second.
Hello, everyone. My name is Isaac Overcast. I'm from the California Academy of Sciences. I'm sorry that I can't be with you live today. Something came up at the last minute. So I'm sending this video instead. My apologies. So I'm really excited to share my work with this group. I'm going to talk to you about leveraging the distribution of genetic variation from ecological communities for conservation and monitoring. So I don't think I need to convince this group that DNA sequence data is critical for conservation and monitoring. The question that I have is, what can be gained by expanding genetic diversity beyond single species? And if there is such a gain, how can we get DNA sequence data for entire communities? Well, the past 10, 15 years or so, metabarcoding has become a really prominent means of collecting this kind of genetic data, where you can go out in the field and sample soil or water or even air these days, and you can extract the DNA and amplify different primers for plants or insects or vertebrates, and then sequence the whole sample and construct presence-absence matrices or relative abundance distributions, or what I'm most interested in is getting genetic diversity data at the scale of the entire community. So what I'm going to talk to you about today is how we can study biodiversity using the community genetic diversity distribution. The community genetic diversity distribution shares a lot of similarity in how it's constructed to the species abundance distribution from the kind of classical ecological metric, where on the left here you see the rank abundance distribution of a community, where you go out and sample the community and you count the number of individuals of each species, and then you just plot the abundances in rank order, and it gives you this characteristic distribution where there are few very abundant species and most species are rare. What I'm really interested in is doing something kind of similar, but using genetic diversity instead of abundances. And so how you can construct the community genetic diversity distribution as go out and sample a community, collect metabarcode data, you determine ASVs computationally, and then you can cluster ASVs. ASVs are individual haplotypes. You cluster the haplotypes by sequence similarity, and then calculate nucleotide diversity within clusters. And you have a kind of genetic profile of the community, where on the right you have this genetic diversity distribution, where this is the most genetically diverse OTU or species, and second most, third most, and so on. So you have this snapshot of community history on a population genetic timescale. What I want to talk to you about is, there are some benefits of the CGD. First, that it's easy to measure relatively, it's robust to sampling, it's independent of taxonomy, it's information rich, and I think it responds on a timescale that I think is useful for conservation and monitoring. Now I'm going to show you a couple of examples of CGD from some empirical data sets. So this is bulk arthropod metabarcode data from Hawaii. Hawaii follows an age gradient where the Big Island is the youngest and Kauai is the oldest, and the age goes this way. Graham and collaborators collected metabarcode data at all these sites. And so I took the metabarcode data and calculated CGD within each site. And now I'm plotting the CGD as a function of substrate age. And so what you can see is, as the substrate age kind of increases, the community genetic diversity distribution increases in a similar kind of characteristic fashion. We can, this isn't just in Hawaii, we can look at arthropod communities in a different system. This is the Canary Islands. In this case, the age gradient goes this way. And I did something similar with the data from Sauces Castellano, where they sampled at all these different sites. And then I constructed the CGD and plotted based on the colored according to age of the substrate, age of the island actually. And you can see a similar kind of pattern of the CGD kind of reflecting the age gradient of the different communities. The CGD also captures signatures of disturbance. So in this case, this is eDNA data from marine samples from Holman 2021. They had paired disturbed intact marine sites. And so I constructed the CGD from some of their paired marine sites. The disturbed are in red, the intact are in blue, and you can see there's this increase of genetic diversity in the intact sites with respect to the paired disturbed sites. The CGD is also sensitive to environmental variation. So this is, again, bulk arthropod metabarcode data from Nogorales et al. 2021, and they collected soil arthropod samples from different habitat types across the montane forests of Cyprus. And each of the points is a sampling site, and so I aggregated the data from all of these points and constructed the CGD and plotted the CGDs according to the dominant habitat type. And you can see that the more widely distributed habitats have increased community genetic diversity with respect to the more restricted habitats. So all these are just, these are very tantalizing patterns. But they're still patterns of the data. So we want to understand these patterns. We need to construct a model. And so how can we model patterns of community genetic diversity? Well, we can start with neutral ecological theory, like the classic MacArthur and Wilson, or neutral theory of biodiversity. And we can join this with a neutral theory of population genetics within species. So there's a joint community population model. I've implemented this model in the massive eco-evolutionary synthesis simulations. We're not going to go into the details of this right now, but essentially it's a linked meta-community, local community population genetic model. And for the purpose of just the rest of this brief presentation, I'm going to talk to you about the model. By default is an ecologically neutral model, but we can also consider non-neutral processes. For example, if species interact with the environment, or an environmental filtering model, or competitively interacting with one another. So we can run some simulations under these three different community assembly dynamics, and we can see that the neutral and non-neutral processes produce distinguishable and different patterns in the community genetic diversity distribution. The simulations are temporally dependent, so we can also run the simulations forward in time, and they also, the simulations recapitulate this kind of age gradient pattern on the right in the simulations as we see in the empirical data on the left. Something that I would be really interested to discuss further in the future, if we get the opportunity, is thinking about how community genetic diversity summary statistics can be potential biodiversity indicators. So if we look, this is the temporal CGD simulations on the left, and we can take summaries of these distributions, like the Shannon diversity. This is the Shannon diversity, or the genetic diversity evenness. So these are two different summaries of the shapes of these distributions, and we can plot them in this two-dimensional space and see that as the communities age, as the simulations increase in age, there's increase in Shannon diversity and genetic diversity evenness, which again, I need to look at more empirical data and do more simulation studies, but I think that this could be useful biodiversity indicators. So yeah, in the kind of short amount of time that I had to share this with you, I hope that I've at least given you some thoughts about how community genetic diversity can be useful for studying conservation and monitoring. It focuses on the whole community instead of individual species, so it captures information at the community scale. It's information rich. It's easy to measure with metabarcode or eDNA data. It's relatively robust to sampling. It's independent of taxonomy, so you don't need a well-curated reference database in order to kind of leverage the community scale genetic data. And it responds on a time scale that's useful for conservation. I was asked to kind of think about some questions that I typically get at the ends of talks, but instead I think what I would like to do is pose questions to the group that I have. So one question that I have is the CGD useful for the genetic composition working group, in the sense that species identity is almost always never known in the absence of a curated reference database? This is a kind of related question. Is a CGD better thought of as a genetic composition or a community composition EBV? I kind of go back and forth about which group might find it most useful. and relevant. And another question that I might have is the CGD, well, it's good for arthropods and in marine invertebrates. Is it potentially useful for vertebrate communities if we can implement eDNA surveys? I'm still not sure. I haven't. It's still an open question. So that's all I have for you. Again, I'm sorry. Thank you for listening. I wish that I was there in person to discuss these things with you further, but I hope that we will be able to chat sometime in the future. Thanks for your attention.
Thanks for sharing, Catherine. Kind of feels a bit weird not being able to throw to Isaac straight away. But I will open this to any quick discussion that anyone might, or point anyone might like to make. I can see Ollie's put something in the comment, and perhaps we can come back to this at the end as well. It's a pretty neat way to capitalize on a lot of eDNA monitoring data, I suspect. Okay, I can see nods but no hands. So how about we move on to our final talk and then we can all chat at the end. In which case I am pleased to welcome Jess da Silva to give our invited talk at the end, which is around building a multi-dimensional biodiversity index with a spotlight on the effective population size genetic diversity indicator. So Jess, we haven't tested this, but do you want to try sharing your screen and we'll see if it behaves.
Okay, can you see that? Yes, perfect, great. Good morning, everyone, or afternoon or evening, depending on where you are. So my talk today is going to focus on just one of the genetic diversity indicators that have been developed, and also how that has been incorporated into a composite metric that aims to give a picture of a country's overall genetic or biodiversity health, sorry. And I'll primarily be focusing on South Africa, but some of the concepts are quite broad. So for years, people have been looking for a single biological indicator to rule them all, a simple metric to effectively quantify the state of biodiversity within a country. And for years, this was debated, but the complexity of biological diversity across its various scales was simply too difficult. Some therefore thought it best to use a top-down approach, but it was deemed too broad, missing out on key items of diversity. So others then felt it best to use a bottom-up approach, but at the time there simply wasn't suitable indicators to measure everything, especially genetic diversity. But even still, biodiversity doesn't really follow a linear pattern. Everything is intertwined, and biodiversity even goes beyond the compositional level into the structural and functional. So there really can there really be an indicator that can encompass all of it? Some people will still debate this, but policy people and decision makers have really been pushing and requesting for it in their hopes to make national and international reporting simpler. And within the past 3 or 5 years or so, there has been a lot of effort put into filling in these information gaps through the development of new indicators or refining existing indicators. And I'll share the developments from the genetic side of the biodiversity spectrum, and then how this ties into the development of a single biodiversity metric. But first, I just wanted to provide some background on the particular indicator for those who might not be fully aware, but I think most of you should be. It's the proportion of populations within species with an effective population size greater than 500. So this is very much a mouthful, so we basically refer to it as the NE500 indicator. And this has become the headline indicator to address Goal A and Target 4 of the Kunming-Montreal Global Biodiversity and Monitoring Frameworks. And just for those who aren't as ingrained as I am in this policy space in the GBF, Goal A, at least the genetic component of it, states that genetic diversity within populations of wild and domesticated species is maintained, safeguarding their adaptive potential. And then in Target 4, this is reiterated, expanded, again, specifically the genetic component section, to maintain and restore the genetic diversity within and between populations of native, wild, and domesticated species to maintain their adaptive potential. And the NE5 indicator was proposed by the Geobon Genetic Composition Working Group as an essential biodiversity variable, and it really helped promote it as a suitable indicator for global biodiversity reporting, as previous frameworks lack this component essentially entirely. And to prove that it was indeed a suitable indicator for the, at that time, upcoming biodiversity strategy, a bunch of researchers got together to test it, along with 2 other genetic indicators. And so the pilot project consisted of 9 countries that varied in biodiversity richness and socioeconomic status. And we were able to assess over 900 species across a variety of taxonomic groups, conservation status, habitat types, and life histories. And this amounted to over 5,000 populations being assessed over a period of just a few months. And of the populations that were assessed, the vast majority did not have effective population size estimates based on actual genetic data. You can see here by this, very small portion. And of the few that did, the information was simply taken from what was publicly available in the literature. Indeed, most of the data used to calculate effective population size was taken from proxy data, specifically census size, either as point estimates or broad ranges. And this was, we thought, a great outcome, as it clearly showed that data, proxy or actual genetic data, was available for a variety of these different countries. Even though it wasn't based on necessarily genetic data, there was good proxy data available. And when we look at the actual indicator values, and just to draw your attention, so 0 here at this end at the left indicates that there are no populations with an effective population size above 500, and 1, all of them are. So when we look at this indicator value, we find that the majority of species had all of their populations below the critical threshold of 500, which was quite worrying. And this was distributed fairly evenly across plants and animal groups. And while it's easy to immediately think that our results are likely an artifact of the species that were chosen, I just wanted to point out that a key selection criteria to increase representativeness and minimize bias was to select species across a range of conservation statuses. And this plot shows that even unthreatened species, so primarily the near threatened and least concerned, had low indicator values below the critical 500 threshold, and so are experiencing genetic erosion. Even they are deemed to be unthreatened, not threatened. So in the end, we compiled overall indicator values for each of the 9 countries, which I've highlighted here in this box. And then these country level indicators, looking at South Africa here, can be disaggregated to get more detailed information on the genetic health or status within taxonomic groups, for example, as shown here on the left, or it could be conservation status, or however you would like to disaggregate it. And the findings of the study were quite significant, as it really helped and is continuing to help countries see how and why they should incorporate genetic diversity assessments in their national monitoring and reporting. Additionally, and at least from a South African perspective, this study really enabled the idea of having a unified biodiversity metric to be reconsidered and developed, because now we have a way of considering all levels of biological diversity. So this takes me to the Multidimensional Biodiversity Index, or MBI for short. And this index was designed with decision makers in mind to assist with biodiversity planning. It measures key aspects of biodiversity related to both nature and people by integrating measures on the state of biodiversity and its contributions to people. And it's fully aligned with the Global Biodiversity Framework, as well as other fora and initiatives. And the hope is that it is or becomes a way of simplifying and commuting biodiversity data. However, this is very much still in a pilot phase and testing, so bear that in mind. I mentioned at the beginning that the MBI is a composite indicator, and so there is the 2 main components, the biodiversity for nature and biodiversity for people, and these are referred to as sub-indices. And I will primarily be focusing on this biodiversity section. And so within the sub-index, there are different dimensions, such as diversity, abundance of species, and functioning of ecosystems. And for each dimension, there are specific biodiversity health objectives that guide the selection of indicators. In this way, the MBI is attempting to capture as much diversity in the structural, compositional, and functional components, and at all three levels of diversity. The development and implementation of the MBI is being led by the United Nations Environment Programme's World Conservation Monitoring Center, that's a mouthful, and is currently being piloted by 4 countries, of which South Africa is one. And to be considered as an indicator for this biodiversity sub-index, the indicators were assessed based on 4 criteria. They had to be broadly representative of biodiversity, so they had to encompass a wide range of taxonomic groups. They should be compatible with other indicators, and so by being scaled from 0 to 1, that would help them be easier to integrate. They needed to have reporting links to major international fora, and they must have suitable data quality, and this was in terms of comprehensive fine-scale national data. So in terms of indicators for the biodiversity section, many of them are actually headline component or complementary indicators under Goal A of the GBF. And in doing so, this index really tries to capture the 3 main focal areas of Goal A. And so within SANBI, the South African National Biodiversity Institute, where I'm based, it's set out to inform policy, and South Africa is really aligned with these CBD global strategies. And so a lot of our work specifically aligns to those strategies. And so a lot of these indicators were what we were incorporating into our everyday work for our national reporting. So what ends up happening is that scores for each indicator are compiled, and then the overall geometric mean is calculated. And so this score over here, 0.42, was taken directly from the overall country score for the NE 500 indicator that was calculated for South Africa in that multinational study. And overall, South Africa has fairly high scores, and within the biodiversity sub-index, biodiversity for nature, the genetic indicator is by far the lowest. And South Africa's overall MBI score is 0.71, which I think is fairly high. This is mainly attributed to very low levels of potential loss of phylogenetic diversity, it's a big slice, low numbers of species at risk of extinction, the taxonomic pie, and that's based on the red list of species, and high intactness of natural areas providing regulatory ecosystem services. And those concerning trends in South Africa's biodiversity health are high levels of loss of genetic diversity, which I've already pointed out, across a range of South African species. And it may be that this indicator underestimates the genetic diversity of South African species because only a small number of species were included in that first assessment. However, the multinational study showed that this was simply not a pattern observed in South Africa. Many countries had even lower levels of the NE 500 indicator. And additionally, the indicator on species population trends, so that's over here in this part of the abundance section, showed declining populations compared to those that are recognized as being at risk of extinction, again, based on the red list of species. So it seemed that the NE 500 indicator and the indicator on population stability could be providing a much wider perspective on species diversity and abundance than other indicators, which is, and so the red list of species is one of South Africa's headline indicators. So that's to differentiate from the GBF headline indicator. And we use it in our national biodiversity reporting and report on it every 5 years. And the combination of the 3 indicators suggests that while the risk of extinction of most species may currently be low, we can expect ongoing population and genetic decline to lead to increases in the risk of extinction in the future. So as part of the pilot study, each indicator was rated. And while the NE5 indicator did really well in biodiversity representativeness, it scored quite poorly in data quality. But it's not exactly the data quality, but rather it's more related to data abundance. So indicators that scored high had representation from the 8 complete taxonomic groups that are assessed and represented, reported on in South Africa's National Biodiversity Assessment. And while our pilot study covered all of these taxonomic groups, it didn't cover them in their entirety. We only assessed 120 species. So I don't really take much offense to the frown, but I am committed to reversing it and already started some efforts to do this. So I'm currently compiling data for complete taxon groups in South Africa, and these include mammals, amphibians, and a cohort of marine species, and this should be completed by the end of next year. And then going forward, I'm looking to complement the taxonomic representation to complement it with the red list of species and include reptiles, birds, and some plant families from 2026 onward. And then I'm hoping to continually build upon this. And so this is for complete taxon representation. And to do this, I've partnered with our red listing team that undertakes reassessments every 5 to 10 years, and as well as new assessments every year, provided there's capacity. And we're going to be incorporating the necessary information needed to calculate the NE500 indicator, but also the other some other indicators, proportion of populations maintained, and genetic monitoring indicator. And we're doing this in a designated population genetic section within each of our regional assessments. And yesterday there was a talk by Julia, work that was being done with the Conservation Genetic Specialist Group of IUCN on defining ESUs and subpopulations. And so ideally that mechanism scorecard will also be incorporated into these evaluations as well. And so going forward, SANBI envisages that this index could potentially be used to monitor changes over time, conduct comparisons of change in space, and even compare the status to different countries. And if anyone is interested in any more of this, since I gave just a brief overview of everything, these are some of the key publications that will help really unpack everything, but also feel free to reach out and I can connect you to many of the right people. And I just want to thank all of the authors that were involved in that multinational study, as well as the SANBI MBI team for making essentially this presentation possible. Thank you.
Thanks, Jess. That was great to see that summary. And it's kind of sobering, but also makes me optimistic that at least by recognizing the problem, we can start to work on it. So does anyone have a question for Jess before we move to the general discussion? I was going to ask you about the taxonomic coverage, but then you answered that at the end.
Too comprehensive.
Jutta, do you want to ask?
Yeah, this is really exciting, this multidimensional indicator. Maybe it is completely unrealistic, at least with the data and the concept we have today, but is there anybody looking into a Bayesian approach of integrating the different levels of diversity into one single outcome? Do you know about that kind of an approach?
So by different levels of diversity, are you talking about ecosystem, species, and genes? Or yes. So the indicator does. So it looks at the red list of ecosystems as an indicator, which South Africa it's another headline indicator for South Africa. And for the species component, it is using the red list of species. And then there's a few genetic components. There's phylogenetic diversity as well as the NE500 indicator. So is that what you had in mind?
Well, actually, well, it is kind of like, yeah, maybe not realistic, but I'm actually thinking about like a Bayesian approach of estimating genetic diversity, like not as a proxy, but really genetic diversity, integrating that then with taxon numbers, an estimate of taxon diversity, and then scale it, like taking that in a hierarchical Bayesian approach higher up into the different ecosystems that are present, different habitats, and running a hierarchical Bayesian approach through all of this to really, yeah.
Yes, I agree. That would be fantastic and an ideal thing. And so I consider this more as a growing, fluid, iterative kind of index, or because South Africa is actually quite good with its genetic information compared to other parts of Africa, but even still, it's by nowhere near as there's nowhere near as much information as perhaps there might be in Europe or North America or Australia even. So I think it's in time, as the data become available, it can help refine it. And I think it can only get better as more information is there. So I think it's something to strive towards. But with other countries piloting this and incorporating how they can do this, that would be really informative. Countries with this information already in hand would be extremely useful.
Yes, and I don't want to I see this as a wonderful approach, and that really, if you can have more countries applying it,
Yes.
it will be just really wonderful. So
yeah, because the 4 countries was just a brief pilot, but the next phase I think would really be to incorporate other countries, seeing what different indicators even that they might have that could be better informing it. So yeah, I 100% agree.
Thanks, Jutta. Peter, do you have a question?
Yeah, Jess, I was wondering how you deal with populations which are over the population estimate threshold, let's say 2,000, but are fragmented, and you don't understand the genetic exchange between these, call them subpopulations, in
Yes.
estimating whether they meet your NE 500 level or not.
Yeah, there's still lots of work to do with the genetic indicators and answering the questions about how to handle that fragmentation. Were they still connected, and should we be considering them as different subpopulations? All of these conversations are happening, and hopefully we'll potentially get a more refined metric on that better captures it. But at the moment, at least I am basing it on heavy expert consultation, people on the ground who have a better idea of what potentially could be separating these populations, subpopulations, however you want to call them, and if there's any form of connectivity. So if there is some form of gene flow, at least one generation, one migrant per generation, whether this is based on natural movement or human mediated, then it would be considered part of a single metapopulation. And it would be that metapopulation that is then evaluated based on that 500 threshold. Does that answer in part your question?
Yeah, no, it does. Thank you. And I think the question that emerges that for a manager, for instance, take the black rhino population of Southern Africa,
Yes.
is what kind of gene flow do we need to augment, or how do we augment gene flow so that we can reach that threshold? Because no single population is above, or no single population is above 200 at the moment. So one needs to do that in order to do it, and that's the management implication and question, really.
And I think you brought up a good point, and it's even just based on standardizing terms. And so I know in the several red list assessments that I've been a part of, even defining terms as subpopulations when they're actually meaning individual farms or sites or private reserves. And that doesn't necessarily really measure what a subpopulation is from a genetic perspective. So we are hopefully working to to standardize some of these terms, and I'm really working with some of the managers and assessors to really understand what exactly they're meaning, because it can have large implications. And you mentioned yesterday a question about the green list, the IUCN green list, and 100%, I think it could also inform this information, how green listing gets, or recovery, species recovery gets, especially with the case of the rhino. It shows that there's been recovery. If it had a green list, it would show that the species has recovered really well. However, the effective population size is what, 20 individuals is 20. So it's extremely low. So it could help inform that side of things. So it's all about integrating all these different bits of knowledge and fora to get the message across.
Thank you. Thanks very much.
Thanks all, thanks Jess. So what I would like to do now is open a few more minutes specifically to the last, well, I was going to say 3 speakers. We can talk about Isaac's talk as well. So just to recap, we've had the case study from Sasha on the developing the genetic strategy for managing fish metapopulations. We've heard about new directions for monitoring genetic diversity using eDNA for community genetic diversity. And then Jess's overview of one metric to rule them all. So if anyone wants to talk or ask another question about that in the next couple of minutes, and then for the last 15, I think what I'd like is just open it to more broader topics. If no one's got specific questions, we can go straight to that part as well. Oh, we have hands. Paul was first.
Oh, thank you. Just very quickly, Jess, how that looks humongous. Like what resources do you need to throw at that?
Okay, yeah, lots. So so as I said, SANBI, the organization I'm with, it really focuses on biodiversity monitoring and reporting. And it has, over the past like 10, 20 years, it's really expanded a lot of its efforts on the red list of species and the red list of ecosystems. And South Africa invests a lot of time and research into getting comprehensive mapping across its oceans, coast, and throughout its interior. So all of this has made all of these indicators possible. So it's not just about personnel, it's about decades of work that has gone into making sure that the data is fine scale enough and of really good quality across a variety of taxonomic groups. So it takes time. So some people say when we present some of this stuff that, oh, but South Africa, it's been doing this for so long and it has this, but we're still quite new on the genetic side, but it's just time and commitment. So countries will have this to varying degrees, but it is possible. And yeah, but it's actually very few people working on it. It's just taken a lot of time, a lot of iterations, and a lot of collaborations internationally to refine the data sets that we have.
Great, thanks.
I guess I can
Sorry,
go.
I lost my mouse. Jess, you've got your hand up, so I assume you're not asking yourself.
Right. It was more for Sasha, but I think it could just be a broad question as well. And it's more curiosity, not based on the actual methods that were proposed. But for the genetically distinct groups that were identified, are there efforts to biobank these unique units to potentially aid in future reintroductions if these genetic lines get lost? And so the talk focused on reintroducing an extinct population, but the genetics of that population potentially wouldn't have been known, or were they known? I think I've missed that. So I was just wondering if efforts within Australia or potentially in other regions is heavily focused on really trying to, once these genetic units are identified, to store some reference material for potential future efforts.
I can start answering that question. So for my garbage, populations, a lot of populations were lost in the last 100 years. So no biobanking. They're also considered to be quite common in populations where they still persist, which is a bit paradoxically. So like when they spawn, for example, they form spawning aggregations, so people, see them. So that is kind of not on the minds of managers yet. But for some other species that are much, much rarer, for example, helmeted honeyeaters or leadbeater's possum, there are efforts now to biobank some of this genetic diversity, even though we are starting to introduce gene flow from other subspecies just to maintain a functionally working population, because then breeding depression really kicks in. So efforts are slow. Museum Victoria now in Victoria, Australia, is starting a culture of collecting cultures, tissue cultures. So they are now doing this, I think, opportunistically. So this is really hard to find funding for this kind of thing. So although they're doing it, it's not like something they intentionally go and do. So it depends on really goodwill of people involved in threatened species to, to do that.
And just yet another legacy of our dear friend Mike Bruford as well, of getting cryobanking sort of ramping up at some sort of scale.
Thanks. Is there any, I mean, we've had a few Australian, I know we're dominated by Australians on this meeting, but does anyone else want to speak to other countries on that topic? I know that the IUCN group has a paper on recommendations for biobanking that is somewhere in review at the moment. So there should be a white paper coming out from the Conservation Genetics Specialist Group soon on that. Broadly applicable, but yeah. Okay, so what I'll do, I've been taking some notes and I'm just going to throw back at everybody. I've been trying to group ideas as they've come up, just to kind of put some things out for discussion. So either in the chat or in the conversations, we've had some questions about training in conservation genetics and how to access training opportunities. We've talked quite a bit about management of populations or ESUs, the importance of local adaptation and gene flow and whether things should be mixed or not. We've talked about the use of genetic diversity data to determine things like provenance of specimens. So I guess value add from the current topics being considered. We've had some discussions around scaling up to large scale monitoring from, species where you know an awful lot about one species to cases where you may know very little about lots of species. The community genetic diversity talk, and then the next steps for indicators, so building on the current work to the multidimensional things. So those are the kind of the dot points I've made on my sheet. I guess I'm just going to throw it open to anyone to either add a new topic or expand on one of those if you have questions while we're here, and thoughts on, what you think this working group could do next. And sorry, Jasmine, I didn't see your hand is up.
I just raised it. Thank you. I was wondering if we could elaborate a bit more on the part of mixing different ESUs or populations that are potentially locally adapted in terms of genetic homogenization, or what are your thoughts about this?
I can see Paul is itching to reply.
Yeah, well, I am, but I also would like to give somebody else a chance to say something. I mean, I can talk about this topic underwater. So. I can give you some, well, two headline things, but one interesting aspect is I've been having a sort of fairly long and detailed conversation with Sean Hoban about this, because there is potential, and this is something that I'd not really been sort of aware of, but with the essential biodiversity indicators, there is actually potential for them to, if they're inappropriately applied or interpreted, there's potential for them to have perverse consequences. So if one of your indicators is many genetically separate populations, and you had a small one that was dwindling to extinction, which is exactly like the helmeted honeyeater that Sasha was talking about, down to 50 individuals, inbred to hell, very, very strong inbreeding depression, like 90% fitness loss through being inbred. So we have now very successfully, happily, and everybody loves it, mixed it with another subspecies, and now it isn't inbred to hell and it's doing better, and there's another population been set up and so on. So depending on your definition and what you measure and whether you're thinking about it or not, it could be interpreted that we had two distinct populations, now we only had one, and that's a loss of biodiversity. Only it isn't, because what we have is the gene pool of the thing that would have gone extinct hasn't gone extinct. So we've actually maintained biodiversity. So that's the kind of relatively novel, I think, thought. At least it was novel for me, and I've been sort of talking to Sean about that quite a lot and sort of unpacking what the various implications might be. He has been very, very clear that he and the group never intended the distinct populations criterion to be inhibitory to genetic rescue, because sometimes genetic rescue is the best thing to do if your goal is to avoid extinction. So, and that's my main point about your question is what I said before, whether you should mix or not. There are, we've written and demonstrated with explicit examples what your thought processes can be, your decision-making tools and so on. Very happy to provide all of that, so you could, if you're not aware of it and you want to have a look at it. But you need an evidence-based approach to make decisions. You decide what it is you're trying to achieve, and then you do the best thing as far as you know for trying to achieve that goal, and monitor, and if you don't like how it's going, then do something a bit different. Jessica has a hand up. Sorry, go on.
Okay, so yeah, I see really depending on the species itself, and if they have populations that are at risk of extinction, then rather, yeah, and otherwise rather keep it separate, I see.
Well, I mean, Sasha's presentation was about proactively mixing different populations to attain sufficient effective population size, which is incredibly aligned with what Jess was talking about. So yes, very dependent on circumstance.
Yeah, I just wanted to reiterate that, because I don't think there ever should be a hard rule, because that could kick everyone in the foot. And there are many examples where mixing or keeping them separate have had unintended negative consequences. So it really is based on the best available information at the time. And I believe the ultimate goal is to prevent extinction. So it really is a case-by-case basis. And I also just wanted to go back to the indicator as well, that there really are, there could be perverse outcomes, especially since NE 500 is a headline indicator. So technically, if you don't report it alongside the complementary indicator, or component indicator, which is portion of populations maintained, you could get a false sense that your country is improving its genetic diversity if you would lose distinct species. So if you don't necessarily have to report on that portion of populations maintained, it could look like your scores are higher if you just lose that really bad, like genetically eroded population. So what could be really good, I'm not sure if it's for this working group, perhaps a project can be proposed. And I know there are some efforts with other groups to think about this, but nothing has been concrete, is to try to see if somehow those indicators can become one, like there could be some sort of measure to more effectively encompass what both are trying to capture, because the intention was that they are reported together and not separately because of the consequences that could be reversed. And then in that fragmentation, if a species or populations become more fragmented, does that mean you have more diversity or more populations? No. So there's all these nuances that need to be sorted out, which would help refine it. So this was a first stab with the indicators, but there's definitely room to grow. And so I'm not sure future work of this group could focus on really teasing those things out and refining this body.
Sorry, I'm too busy taking notes here. And then I remembered it's all being recorded anyway. I think that Jutta's hand was up next and then Sasha's.
So I wanted to shift gears. So Alexandra, if you have something in response, you should go first.
Okay, thank you, Jutta. My kind of thought on how indicators can be joined together. I was wondering if having a sort of GIS point for each estimate would help. For example, each estimate is actually linked to the place where which was measured, because then it would be not just kind of a population, it would be a particular spot that would be either linked or not linked, and then it would be more objective. The question is, how do you then provide a cumulative estimate? Maybe this will be something like community estimates, something like what we've heard from Isaac, maybe something more species specific, but I was just wondering if that will allow avoiding that kind of issue of removing one population just to improve the, evidence of being good. I'm sure you guys have thought about this before.
Can I quickly respond to that? It has been a thought. It hasn't been articulated. So even with a lot of these, the multidimensional indices and working with a Namibian team to also try to quantify indicators across, that they all want spatial elements because it's easier to present indicators or values for biodiversity if you can show like a policymaker a map and show where it's high and where it's low. So there's been thoughts of that. So yes, I think that could be a really great way to somehow do it, but it hasn't been at all thought out.
Mm-hmm.
It also does have the allied complexities that as soon as you start doing things that are ex situ, then what does that mean to your spatially explicit point? So yeah, I mean, I think what Jess's basic framing around, we just need to make sure that the indicators are being used for the purpose for what it was they were trying to capture is the sensible thing. So the whole point of the genetically distinct populations was didn't want to be losing lineages with unique variation. If your way to not lose them is to mix them with something else, then they should still count as having been not lost.
Thanks, Paul. Jutta, your hand has gone down. Did you want to say something, or should we go to Peter?
Yes, the topics that I wanted to raise, we talked about how to fund our efforts. That is kind of what I wanted to raise, but we are at the end of time, so I'm not quite sure if I should throw this in. So.
It's a good question. It's always a relevant question. Did you mean
my question that I have and that has been in the back of my mind for a while is Have people thought of connecting our efforts with the UN system on environmental economic accounting? And I also know UNEP VCMC recently had a similar accounting approach that I came across. And I know from the CR, there is interest in species level and genetic level genomic data. I'm wondering if here from this group, there are already connections in that direction, because I know my latest information is that they're currently actually working on integrating species level and lower diversity into their statistical framework. And it could be, so the idea is basically, the more data we provide, the more money we could get. And there are ideas how one could start such a system where a big commercial business or so might step in and help to jumpstart this kind of a system. Because like everything we talk about is hugely work intense, and we need so much more development. So maybe this could be an approach to funding. Thanks.
Thanks. Yeah, so that's a great point. I don't think I've come across it in this context, but I think it was in a different context talking about marine monitoring. So I've made a note, and maybe one of the others has looked into it, but I'll follow it up. Yeah. Peter, did you also have a point you wanted to make on one of those conversations?
Yeah, it was very much more about the augmentation and the mixing of distinct subpopulations. Has anybody encountered a situation where there's a distinct gradient in allelic diversity? For instance, in Rana, and I go back to the Rana example, there's a clear gradient of the loss of alleles from, let's say, East African populations down into Southern African populations, which is probably related to the dispersal, the evolutionary dispersal of the species. And then how one deals with now trying to reconstruct some kind of a gradient that used to exist. I was interested to see whether that had been encountered in the fish data, for instance, where down the east coast of Australia, you have a potentially a similar gradient of change that one would then modify your supplementation between populations with nearest neighbor populations as opposed to ones from north to south and so on. I'm just interested if that kind of problem has been tackled at all.
Well, I can speak about, I think you mean genetic diversity that is adaptive, but please let me know if not, because if it's not adaptive but neutral, then why do we need to restore the gradient? Like, this is kind of non-consequential. We want to worry about species, not gradient in genetic diversity. But if it is adaptive genetic diversity that you're talking about, then there may be, a case for not moving individuals from, populations that are too divergent. The current thinking about this is that species were fluid in historical contents. They would be, diverging and then merging again when climate was changing. There was, sometimes they diverged to the point then when they meet, they didn't interbreed, sometimes they did. So gene exchange is kind of a normal part of evolutionary process, and it fuels adaptation. And in some cases, even when species are diverged, some gene flow still happens and some adaptation still can be obtained from, closely related species. in the context of climate change, can we really say that any of the currently existing populations, threatened populations, live in a climate to which they are adapted to? I think probably not, because the environment changed so much. Climate has changed, but altogether, with all of these anthropogenic threats and novel pest species and diseases, like the climate is constantly changing, environment constantly changing. So the variation that species got stuck with is not necessarily making them the best adapted to the location. So, and we as managers of the genetic diversity cannot really predict which population can be a good source. So why don't we try this and that and see what is working best? Because selection is smarter than us, and it should be making a good choice for the population. So our job is just to provide enough effective population, sufficient effective population size for the population to work properly, for the selection to work properly, and then let selection to take its course. And that thinking is now probably most applicable to the marine environment, where gene flow is relatively high, but still insufficient to evolve new adaptations. So there is now, we're thinking now, but maybe we need to move things not from the neighboring population, but, from neighboring and some populations that are away, or maybe even, more extreme. So there is a bunch of variation that is available, not just a limited section. How do we know what's best? We should experiment and see what works, because we don't have time.
I think it's a bit easier to experiment with fish than it is with rhino.
It is. Rhino is like, we should never be in a situation as well with rhino.
Yeah, thank you.
Thanks, everyone. I'm conscious we're now past our stated end time, so whilst I'm happy to keep chatting a bit, I also realise everyone's probably got other things to do, especially some of the Aussies who probably won't go home if they're not home already. So I will thank everyone. I know we've lost quite a few people already with the time zones, but thanks to everyone, especially our speakers, but everyone for joining in and listening and being part of these discussions. As soon as I know, well, someone will communicate had access recordings. I haven't seen the recordings yet from the first session, so there will be a way to look at these and share these talks. And we will try to come up with a summary of some sort that we can share with people as well. But definitely very keen to keep getting ideas. And if anyone is really keen to be more involved in making things happen, there's 3 of us who kind of find time to do this. So we're very happy to have help. If you've got a burning idea that you want to make happen, please help us. And we'll happily take suggestions. Yeah, thanks all.
Thank you, Anna. Thank you, everyone.
Thanks, everyone. See you.
I'll see you all around. Thanks, Catherine, especially for being the logistical rock.
Thank you.
Bye, everyone.